Hello everyone, welcome back at Basics of Biology. In this video series, we are discussing about enzymes for DNA manipulation. The enzymes used in recombinant DNA technology fall into four broad categories, depending upon the type of reaction they catalyze. In our previous videos, we discussed about nucleases that degrade the nucleic acids and template-dependent polymerases that are used to synthesize new polynucleotides complementary to an existing DNA or RNA template, and end modification enzymes that make changes to the ends of DNA molecule. Now in this video we will discuss about ligases. Ligases are a group of enzymes that catalyze the binding of two molecules. An example is a DNA ligases that link two fragments of DNA by forming a phosphodiester bond. These are also known as molecular glues that are involved in attachment or joining of the DNA fragments together. You might have also heard about these enzymes in DNA replication. Ligases is required for the ligation of Okazaki fragments during lagging strand DNA synthesis as well as for several DNA repair pathways. Now the question is, what is DNA ligase used for? In the cells and in the lab, enzymes called ligases are used to join fragments of DNA together. Only DNA fragments that have matching complementary ends can be joined by ligation. Now, what is the biological significance of DNA ligase in a cell? DNA ligases play an essential role in maintaining genomic integrity by joining breaks in the phosphodiester backbone of DNA that occurs during replication and recombination, and as a consequence of DNA damage and its repair, three human genes, ligase gene 1, ligase gene 3, and LIG4, encode ATP-dependent DNA ligases. Ligases are classified into 6 subclasses: ligases forming carbon-oxygen bonds, ligases forming carbon-sulfur bonds, ligases forming carbon-nitrogen bonds, and ligases forming carbon-carbon bonds, ligases forming phosphoric ester bonds, and ligases forming nitrogen metal bonds. Having enzyme commission number 6.1, 6.2, 6.3, 6.4, 6.5, and 6.6 respectively. Now, what is the purpose of DNA ligation? Ligation of DNA is a critical step in many modern molecular biology workflows. The sealing of nicks between adjacent residues of a single-stranded break on a double-stranded substrate and the joining of double-stranded breaks are enzymatically catalyzed by DNA ligases. DNA ligases that are commonly used in cloning experiments are those obtained from E. coli or from the bacteriophage T4. This was all about DNA ligases. Thank you for watching this video. Please like and subscribe to this channel.